Hello, welcome to Regulatory Affairs 101. I am Nancy. As the Alzheimer's Association International Conference, AAIC 2026, begins this week in London, we are seeing a significant shift in Alzheimer's drug development that has direct regulatory implications. For years, the field has centered on amyloid, but this year's meeting is highlighting a clear movement toward multi-modal, stage-specific therapeutic strategies with tau-targeted programs gaining prominence alongside established amyloid approaches. The treatment landscape is evolving quickly. We are moving beyond single pathway intervention toward integrated frameworks that combine biomarkers, precision diagnostics, and thoughtfully designed combination regimens across the disease continuum. In this video, we'll review the scientific rationale behind tau and amyloid therapies, the regulatory expectations shaping their development, and how both modalities fit into the emerging Alzheimer's treatment paradigm. Let's begin with the broader R&D landscape shift. The field is clearly transitioning from single target to multimodal therapeutics. AAIC 2026 reinforced that Alzheimer's research is moving beyond amyloid-dominant development. While amyloid therapies remain foundational, they are no longer viewed as sufficient on their own. Tau-targeted therapies are gaining momentum because tau pathology correlates more directly with clinical decline, neuronal dysfunction, and disease severity. At the same time, diagnostics, especially plasma biomarkers and advanced imaging, are enabling early and more precise intervention, which regulators increasingly expect. This shift sets the stage for the rise of tau-targeted programs. This slide summarizes the key scientific and regulatory themes emerging from day one of AAIC 2026 this week. The opening sessions emphasize continued advancement in diagnostic and prognostic biomarkers, supporting earlier detection, disease staging, and regulatory-aligned endpoint strategies. Presenters also highlighted new findings in genetic epidemiology across diverse populations, reinforcing the regulatory expectation for representative enrollment in global Alzheimer's programs. Additional sessions focused on lifestyle and prevention research, as well as emerging CNS drug delivery technologies, both of which may influence future therapeutic development pathways. Collectively, these themes signal a clear emphasis on innovation, inclusivity, and methodological rigor, providing a scientific backdrop that aligns with evolving regulatory expectations for next-generation Alzheimer's therapies. These day one themes provide important scientific and regulatory context, and they directly reinforce why tau-targeted therapies are gaining prominence in the current Alzheimer's development landscape. Tau has emerged as the next major frontier in Alzheimer's drug development. Multiple tau programs are advancing into mid-stage clinical development, with select candidates progressing further. These programs target tau tangles, intracellular aggregates, strongly associated with cognitive decline. Biomarker innovation is accelerating this shift. Tools like p-tau 217, tau PET imaging, and neurodegeneration markers allow early detection and more precise patient stratification. And importantly, tau therapies are increasingly viewed as essential components of combination therapy paradigms, addressing downstream neurodegeneration that amyloid therapies alone cannot fully modify. Amyloid therapies remain foundational, especially for early symptomatic and prodromal Alzheimer's disease. We now have first-in-class approvals, Leqembi and Kisunla, supported by FDA review materials, clinical trial data, and expanding real-world evidence. However, safety optimization remains a central regulatory focus. ARIA risk management, particularly in APOE4 carriers, is a key requirement in both labeling and clinical practice. Sponsors continue refining dosing strategies, titration schedules, and monitoring protocols to improve safety and patient experience. Amyloid therapies are not going away, they are evolving. Diagnostics are transforming Alzheimer's drug development. Plasma biomarkers such as p-tau 217 are improving accessibility and scalability for early detection. Amyloid PET and MRI remain essential for safety monitoring, particularly for ARIA detection in anti-amyloid monoclonal antibody trials. Regulators increasingly expect biomarker integration in trial design, enrollment criteria, and evidentiary packages. This diagnostic evolution supports stage-specific therapy selection and future combination strategies. Let's compare tau-targeted and amyloid-targeted therapeutic strategies across key dimensions. Tau therapies address intracellular tau tangles, while amyloid therapies target extracellular amyloid beta plaques. Tau pathology correlates strongly with clinical decline, whereas amyloid represents an early initiating event in Alzheimer's disease. Tau modalities include anti-tau monoclonal antibodies, ASOs, aggregation inhibitors, and investigational vaccines. Amyloid modalities include anti-amyloid monoclonal antibodies, historical secretase modulators, and vaccines. Tau therapies are primary mid-stage with emerging early-stage exploration. Amyloid therapies are most effective in early symptomatic or prodromal disease. Safety profiles differ significantly. Tau therapies show fewer ARIA concerns, while amyloid therapies require MRI-based ARI monitoring. Regulatory outlook. Tau therapies represent the next wave of disease-modifying candidates, while amyloid therapies benefit from established precedent and post-marketing evidence. Regulators expect biomarker clinical correlation for tau programs, especially given their downstream mechanisms. For amyloid therapies, risk-based patient selection, including APOE4 considerations, is essential. Longitudinal safety and real-world evidence are required across both modalities. Combination therapies framework will likely require adaptive trial designs, flexible enrollment strategies, and integrated biomarker endpoints. The future Alzheimer's treatment paradigm is stage-specific and multi-target. Amyloid clearance will remain central for early disease modification. Tau suppressions will be critical for slowing functional decline and addressing neurodegeneration. Emerging adjunct targets such as neuroinflammation and synaptic protection will round out combination regimens. All of this converges toward precision diagnostics and integrated therapeutic strategies. This slide outlines the key clinical development implications for next-generation Alzheimer's therapies. Adaptive trial designs are increasingly necessary, allowing sponsors to refine dosing, adjust cohorts, and incorporate interim biomarker data while maintaining regulatory compliance. Enrollment must be biomarker-driven, using validated tools such as amyloid PET, p-tau217, or tau PET to confirm diagnosis and ensure stage-appropriate patient selection. Safety monitoring remains substantial, with MRI required for ARIA surveillance in anti-amyloid programs, and tau PET or neurodegeneration markers supporting progression assessment in tau-targeted trials. Together, these elements define the modern regulatory framework for Alzheimer's clinical development. To summarize, tau therapies are rising as the next major wave of disease-modifying candidates. Amyloid therapies remain essential but require optimized safety management. Biomarker innovation is reshaping clinical development and regulatory review. The future of Alzheimer's treatment is multimodal, stage-specific, and biomarker-driven. Thank you for watching. If you like the content of this video, please like, subscribe, and comment below. Have a wonderful day.